This headset's messed up. So just do, okay. This headset is messed up. So, okay, now we go through the labs and make sure to get biomes link, duh, what I was trying to do here. So go to biome and then we click biome there. Okay, all right. Now let's go to Gilbert lab, microbiome. What were they working on? SARS-CoV-2. Okay. Biome. Wait, what is this about longitudinal discovery? Survey of matter. That's not about aging though. Okay. I don't think they do aging research. Built into environmental. Immunization. By the way, this is going to be a really slow video or just a session in general because it's going to be, I'm going to record everything basically. So it's going to be long form. I guess nerve. Okay, that's not aging. Rat microbiome. Okay, so Gilbert does not do aging. Franceschi does aging. Franceschi lab microbiome. Now I have to find his lab. I'm not even sure. I'm not sure if his lab is online. Maybe we can find a link from here. Francesco. No, that's Francesco. Where's Claudio Francesco? Claudio Franceschi. It was University of Bologna. Bologna. University of Bologna. Oh wow, Sir Escherton. Okay. Okay. I think he just doesn't have a lab. So we'll just do the next thing and we'll do his research kit. No, he's got to have, what is this? What is this? What is this? This is nothing. No, that's nothing. Well, okay, fine. Okay. We'll just do his ResearchGate profile for now, and we will try to find if you find a link to his lab. Okay, so Petrosino. So Claudio Franceschi, he is the one who came up with the term inflammaging. He's also quite old at this point, so it's not a wonder. Petrosino. Should also change this to be Franceschi since I can't find his lab. Claudio. Okay. Petrosino microbiome. Age, genomics. Of course, the problem here is that it could be aging. Nope. Okay, I'm pretty sure he's just into the core science. Okay. So we'll remove Petrosino, Wine Cove, microbiome. Oh, that's right. He's in Magnitude Biosciences now. So we can actually move him. He is at Durham University, though. We'll put him as both. I think he's at Durham. I think maybe he changed or something. I don't know. Well, we'll put this one in for now. And then we'll put Magnitude Biosciences here. And we will get I think it's still in like stealth mode or something. So we'll just do their LinkedIn for now. And unless, I mean, Magnitude Biosciences. Oh, never mind. See, all it takes is a Google search. All right. So Magnitude Biosciences. Okay. And I'll turn this one light gray. Okay. Sonnenberg. Lab microbiome. Okay, research. Carbohydrates, spatial organization of gut microbiome, pathogens, synthetic biology, dynamics. Okay, I don't think they're doing aging, which is, you know, a shame because I think that they're like one of the bigger labs. I'm not sure in the space, but it is what it is. Okay, so composition. So we'll remove Sonnenberg. Nice icon. Okay. Magnitude Biosciences. Got that. So we did Wine Cove and Claudio. Ginniger Lab. I know Ginniger. Ginniger Microbiome. He is. No, Ginniger Microbiome. Edward Ginniger. Edward. I don't think he has like a specific. I think he might have a lab or something, but this is the best one that I could find before. So, and while it's not here, like he's about on axons and stuff like that, he published a paper and it was like a one-off paper and I talked to him about it. But basically it was about microbiome and flies and how you could if you eliminate the microbiome and flies, like a bunch of the pathologies go away or something like that. Okay, so Knight's lab. I know. Knight lab, UCSD. Research. Microbiome for forensic. Environmental, animals. Nothing about aging. Human associated, intervention, stable over time. Stable over time doesn't necessarily constitute aging. Research. Different disease and conditions. To dietary, antibiotic, microbial. Transfecal plant transplants. American Gut Project, that was pretty big. A Human Microbiome Project is also pretty big. And whilst they seem important. Oh, in addition to supplying US variables, which such as age and information on exercise. Does this count? Sure, why not? At least they mentioned it, right? That's what's important. Okay. So we're gonna put night lab. Link. Okay. Oh wait, was there two different? I think there might have been two different ones. I think there might have been like a night and then nights. Nights lab. So there's nights and there's night. All right. Well, let's see here. See example projects here. University of Minnesota. Per ship, bioinformatics, Shogun. First, I think they're just making tools. And if they're making tools, it is important. But does it constitute aging? I don't see it mentioned anywhere. Okay. All right. Okay, so Night Lab, Blind Cove. Oh, Snyder. Snyder Lab Microbiome. He is doing aging. And so what's their research? Okay. He is looking at, I'm pretty sure he's looking at microbiome because I talked to him and he was looking at microbiome. And I think IPOP does microbiome stuff. Transcriptome, methylome, bacteria, microorganisms in the gut and skin. And he's doing a lot of aging research. Okay, so we will go back to Snyder Lab. And we will put that in the description. Okay, there we go. All right, labs and companies is done. If you know of any labs or companies that are working on the microbiome and aging, the combination of the two, then feel free to let me know because that's what I think the crowdsourced part of this whole thing is going to be really great at identifying the presence of things. Okay, now let's get into the actual meat of this, which is going to be how do we go through and justify the existence of every single one of these things? Okay. This is gonna be tedious, but we are going to go to Science Direct. And we're going to look at these. What we're looking for right now is the origin of microbiome. So we're going to go to microbiome and we're going to try to find the origin paper for when it was like discovered. And that means going back to 98. Fluorescent sensor, the sub 99. 2001. Laser 2002. Host microbe interface. And we're going to need to view the PDF. So we're going to need to go to Sci-Hub. And we're going to go to microbiome. It's not mentioned anywhere. Okay. So going to go to 2003. Host microbial pursuit. Probiotics, 2004. We'll remove all the rest of these just to like make it a little less confusing. Strepto, symbiont. You know what? This is where Wikipedia actually comes into play here. Define the human microbiome. Maybe we don't need Wikipedia. Let's go to this. Let's go to SciHub and let us find microbiome. So 1, 2, and 3, we're going to go to references, the Human Microbiome Project, that's 2007, 2009, 2010. Okay, Human Microbiome Project, it's going to be Nature. Turnbow. That's going to be PDF. Okay. Let's look at the references and see if there's anything that, okay, distal gut microbiome, 2006, theory of island biogeography. Let's do Control F for microbiome, and we'll go through the references and we'll see if it doesn't, oh no. Oh no. Oh wait, there we go. Okay, 2007, 2005, 2006, 2006. Okay, extending our view of self, human microbiomes, national of that. Oh God, page not found. It's been deleted. I guess I'll go just to the 2006 one, metagenomic analysis of the human distal gut microbiome. What we're looking for here is the definition of the term microbiome and when it was originated. And you know what? Might as well just check Wikipedia. Wikipedia micro, because maybe they just did this already, and that's fine. History. Etymology. There we go. See, look at that. Easy peasy. Bioconcepts. 59. Microbiome. What? God damn it. I hate you, Wikipedia. Why don't you have a, okay, the Scientific Monthly? Nope. Microbiome secret. Okay. Gotta go find the thing. Prozoa as indicators of pollution, Scientific Monthly. Semantic Scholar, there we go. No paper link, but God damn it. How do we get this? How do we see the actual paper? It was 74, 7 to 9, 74. 74, 79. Okay, here we go. Here we go. Here we go. Da-da-da-da-da. No, oh my God. Doesn't have a DOI. Okay, fine. We'll just have to use this then. Okay. So this will be the origin. That'll be first, and then we'll do defining the microbiome. I think I saw a paper about that somewhere. Define the microbiome. Where was it? Oh, rejuvenating the human gut microbiome. What's this? Oh, great. That's fantastic. Okay, well, for now, we'll do defining the human microbiome. Looks like, is that Rob Knight of Knight Lab? That's Northwestern. That's UCSD. And who is here? Oh, okay. So he is currently at UCSD. And we'll use this one. And we'll go this. I'll go to Sci-Hub. I'm sure that we will find what we're looking for. Technology trees, and we'll go to folder microbiome. Defining the human microbiome. Since we can't cite Sci-Hub for this, we'll do this instead. Okay, so we're going to go to microbiome, and we'll come down 2 lines, and we'll put the second one. It looks like it saved it. And that's a good start. Obviously, there's going to be like so many of them, but let's do one for microbiome and aging. At least one. This is the Wilmansky. So whose lab was this? Who is the primary contact? We're gonna have to look into this. Link, prozoa, query. Okay, got microbiome patterns. Price. Wait, what? Gut microbiome pattern reflects healthy aging and predicts survival in humans. Okay, and we can actually link directly to the paper here because it is public. That's fine. And now, okay. Gut microbiomes becoming increasingly unique to individuals with age. Leverage The older age, helping to show continued microbial towards unique drift is less absent in healthy individuals. Helping to depletion of core genera across most bacteria. The identified microbiome pattern of healthy aging is characterized by a depletion of core genera found across most humans, primary bacteroids, or having a low gut microbiome predicts decreased survival in a 4-year follow-up. By how much? So let's go to bacteroids. To characterize bacteroides. We're looking for the results section, and we're looking for, okay, bacteria. Okay, so cohorts, weakness score. To fall away from my bacteria. More regenerable owing to firmicutes. They're talking about stuff. Where's the actual numbers? That's discussion. This is probably one of those papers where the results are at the end of the paper. Maybe. Covariates. Males. Touching uniqueness across cohorts. Bacteroids. Spearmint. Okay. Hopefully it's still recording correctly. Is it? Is it recording? Okay, I think it's recording. God, I hope it's recording. Okay. Great. I have to do these like coefficient tables. So how did they measure health here? They measured health here by walk speed, and what else? This is all what references, and tables are down here. How'd they measure health? That's the big part. Okay, minus decades of human life. Results. Introduction. Oh, the results were first. Deeply core is the wellness study and osteoporotic fractures in men. Validation cohort. Baseline data. Data diversity, blah, blah, blah. Oh, great. They're using all sorts of jargon here. Dissimilar, given all of the microbiome measuring uniqueness. So everyone gets more unique with age, with healthy aging. Is that their? See, I don't know to what degree, and it was consistent across for gut microbiome. Okay. And 2 different microorganisms. Rotator analysis using additional. So figure 2B and extended data 3. Extended data figure 1, figure 2, figure 3. So Association between age and gut microbiome uniqueness across cohorts. What? God, what is this? Age, beta coefficient, and corresponding p-values from oil smiles predicting breakers, uniqueness, and genus level in the American Gut Project, and 2 vendors in the Arabical. Analysis was performed on all samples and repeated on a subset of samples at available sex via my data. So was this p-values of just what, greater than 0.5? For what? Oh my God, this is so obfuscated. Spearmint correlations and diversity for gut microbiome. I'm not even going to pretend to understand what this table is actually saying here. So we'll do, oh, they just looked at like a whole bunch of other things as well. This is unusable. All right. Yeah, this is not usable. To me anyway. It's like it's buried between too many layers, beneath too many layers of like information, what's it called, protocols. So let's go to microbiome and aging again. And we will look at a couple of these other ones and hopefully find something that is a little bit more accessible. So full text download PDF, and there's no PDF, so we have to go and find the DUI and go to Sunhub. Okay, microbiome and aging. Of course, it's at the center of everything. The center microbiome is okay, whatever. So they think that it's involved in literally every single hallmark of aging. Okay, sure. Let's see here. So the holobionts, microbiota dysbiosis. So they claim to have that this is a hallmark. And you know what? The hallmarks are really badly organized anyway. So I would agree that adding in a microbiome thing would be a really good idea. Antibiotics, metformin. Okay, so here we have a really great, okay, this is really great. So what we're going to do here is we're going to, well, we can't link to this one. I mean, we can't link it to it directly, but this is the Cabrero. Okay, this is a really great system of keywords. So we're going to put this and we're going to put that into the tech tree. And we're going to pull it down. And now we have a couple other things. So let's take a look at what they were saying. Antibiotics, metformin is involved here. So we're going to have antibiotics, and actually isn't metformin an antibiotic? Because, oh wait, I already have that here. So we have antibiotics. I think metformin is an antibiotic. Kanamycin, rapamycin, metformin. I think metformin is an antibiotic. Let's look it up and spend like 1 second. Metformin antibiotic. Potential antimicrobial agent. It's commonly used anti-diabetic medication. It's multi-drug. It's a potential antibiotic. And multi-drug resistant bacteria. So huge reference to spike, blah blah blah. Positive. So strategies, resistance. So on the hand, yeah, resistant bacteria means non-antibiotic antimicrobials. These medicines not conveyed can exhibit are not called non-antibiotics. So I think that what they're saying here is metformin is a antimicrobial, but not an actual antibiotic. This is evidence for it, regardless. So we will what we'll do here is we will take metformin and detach it, and we'll put in antimicrobials. Microbials. And we'll put metformin inside here. And this becomes an intervention for the microbiome. And the so the first thing is to justify metformin as an antimicrobial. So we will put that link to that paper in the description. And then we will we'll go back, and the question is, see, we can't put it in the link because the link is going to be the connection between metformin and antimicrobials and microbiome interventions of aging. And this is just metformin as an antimicrobial. It doesn't actually describe how you use it to target aging, the mechanism of why targeting the microbiome with metformin would impact aging. Which I had this concept a while ago of like, I think metformin is an antibiotic, but clearly I didn't know that it was not an antibiotic, but acts like one. And that you could use it, that maybe part of the interaction of metformin was microbiome stuff. But anyway, let's move on for now. So we have antibiotics, then antimicrobials. Where's that paper? Here. Okay. We have metformin, high fiber diet. So high fiber diet is going to be in dietary control. And we're going to put in dietary, and we're going to do fiber. We're going to do here's the intervention section of the diet. So we're going to put high fiber diet, and we're going to do that as an intervention. I'm going to get a new headset before I do the next session. This is ridiculous. But okay, so we have a high fiber diet, and that's going to be targeted towards the microbiome. And our justification, maybe we can find a better justification. Oh, high fiber. So maybe we can find a better justification. High fiber diet is, so talking about fiber rich diet, and maybe that's fibers, for example, that's talking about amyloid fibers. So this is the citation 118. 118 is crosstalk between messages from bugs. Oh God, more rabbit holes to go down. Okay. So we're gonna do crosstalk between the microbiome. And we are going to, what we're looking for is a more specific reference for the microbiome. So PDF here. And we're going to look for fiber. What? He doesn't even mention fiber in this whole paper. See, they said that a high fiber diet can, what did they say here? They said high fiber. They said this, okay, these bacteria production is favored by a fiber-rich diet. And we have here the paper that they're referencing, and it doesn't mention fiber at all in the entire paper. That's impossible. No, I don't mean it's impossible that the other paper got it wrong. That happens all the time. But what's impossible is that this is a paper about the microbiome and they don't mention fiber. That just seems like a failure of Control F. There's vitamins. There's 3 strains, folic acid, generate folates. Maybe they're using. God damn, I hate this headset. Okay. Maybe they're using. Dropped it too many times. Dropped the headset too many times. Microbiome histone modification. Microbiome signals and host signals. I mean, holy crap, this is really a lot of really interesting information, but okay. So we're gonna put it seems like a high fiber diet. It makes sense, but the fact that there's no I don't want to go digging for that at this point in time. Although I guess this is the time to go digging for that kind of stuff. But still, we cannot put a high fiber diet. I know that's an intervention. Like I know people think about it, but there's no link. So if you can find a reference, an actual reference for high fiber diet inducing microbiome, I mean, I'm sure that we can find it. But the fact that this paper doesn't even have the right reference is annoying. Western diet being a terrible thing, but that's a decision. So Western diet is not it might impact aging, but it's not a root cause of aging. Do you understand? It's not like people will age even if they don't eat the Western diet. So it's more of a political decision or like a cultural decision and it belongs in the domain of cultural decisions in terms of the solution and the fixing of it rather than being an aging solution. Like you wouldn't study the Western diet if you're studying aging because it's a culture thing. You just decide not to eat the Western diet and then problem solved. It's not difficult. Caloric restriction. Okay. Caloric restriction. Let's see if caloric restriction. So they are going to use CR. Oh, great. But that's caloric restriction. Microbiome from both conditions, often caloric restriction as well intervention that promotes longevity, improves health across species. 93, that must be just about okay. It's actually not that well known. Oh no. Oh no. Okay. Stop. Okay. So we're going to go back up to an overlap between lifespan CR and the microbiome of both rats and mice has been previously reported. Both CR and ablation of the microbiome increased median lifespan of mice and rats. Oh, they're probably talking about Ginniger's stuff. 138, 134, 38. 134. Snyder. Huh. Your diet restricted, germ-free, and conventional gluten. So ablation was there. That was before, it was way before Ginniger. So they already did this, and then Ginniger followed up on it in flies. Okay. Immunological function of food restricted, germ-free, and specific pathogen-free mice. So we're gonna do this, and I'm hoping that they're doing aging here, but it might not be. Mainframe. Attained longest survival rate. Stimulation index. Of course, no graphs about survival. Mitogenesis of spleen cells. Show stimulation. And also this is a calorie restriction back in the day when they don't, like full fed means ad libitum. Is that it? Responses on of aging GF mice. GF is germ-free mice. Gordon reported that germ-free mice outlived conventional mice, and unique characteristics of the germ-free state. So, early life. Okay, so for this study was they were looking at the caloric restriction. So germ-free mice live longer than normal mice, but then within the domain of germ-free mice, they were looking at caloric restriction being either better or worse for them. And we got onto this track by looking for evidence of calorie restriction as an impactor of the microbiome. What we're gonna do here is instead we're gonna go to. I don't think that particular paper was gonna be helpful, but we could go back to up, down. Okay. Did not have an additive effect, so there's an overlap mechanism in regulation of host longevity. In fact, many studies of mice have shown that positive CR-induced metabolic effects on the animal are influenced by remodeling of the gut microbiota. So what they're doing is here they're claiming that calorie restriction is actually explained by the microbiome shifts. CR actively suppresses lipopolysaccharide biosynthesis by microbial, leading regulation of the immune system by TLR4, dependent mechanism, in particular transplantation of and reduces weight gain. So microbiome, functional gut microbiome remodeling constitutes to the caloric restriction induced metabolic improvements. This is a good thing to throw in there. So the idea is microbiome. How do we show this? We have intermittent time restricted feeding, caloric restriction, have protein restriction here. Maybe I'll create a child, I'll put caloric restriction in here. And it's gonna be complicated because caloric restriction actually is a catch-all for other things. Like low, well, but we'll put caloric restriction to the microbiome as an intervention. And we will do, so we'll do this as like that, and then we'll do the paper here. Where was it? Functional gut improvements, this paper. Well, induced. Where is the PDF? Where's the PDF? Come on, guys, get your act together. Okay. Save as microbiome. Functional gut microbiota remodeling contributes caloric induced metabolic. Okay. So toll-like receptor, lipopolysaccharide. So that's going to go into the immune system. So this paper, and we're going to go back to cite the public version of the paper in this. So this intervention is going to have this description, and it's going to come from here to the microbiome, and then from microbiome, it's going to go to the immune system, which is not built out yet, but we'll create a child node here. We will go to make sure it contribute to the serum lipopolysaccharide. I don't think we're going to get to the LPS level of specificity, but let's have the innate immune response as a section within the immune system. Oh, I already created a node. Innate immune response. And we'll put this, oh, sorry, whoops. That's not for the justifying the innate immune response. That is justifying the microbiome impacting the innate immune response. So here we go, description there, justification. We'll put this into there. So now we have caloric restriction comes over, affects the microbiome, comes over to affect the innate immune response. And the innate immune response is probably an intervention somewhere. This thing we can, okay, it's fine. It's fine for now. Okay. You see how slow this is? But this is how long it takes if you want to actually dive into things. So I think this is okay for now. It's just Pat Browning. See, and then what needs to happen is that somebody within the field of microbiome needs to come in and be like, oh yeah, you know, this study is good or bad, depending on whatever. But we might as well look up Salvatore Fabiano and see if they're doing age research. Fabiano lab microbiome. Center of Summertime in Geneva. Do I recognize any of these names? No. But the lab here, okay, so let's just go through as an example of like what, okay, so Trajkovsky lab. That's games. Microbiome. Is he doing aging? Age. Nope, he is not doing age. Okay. It's mostly just like a metabolic diabetes type of research. All right, that's fine. I mean, that's necessary. But it's not aging, so we can't throw that in there. Okay, anyway, so we have that, and it constitutes an innate immune response. But the thing is that the immune response is affecting the microbiome. But is it the innate immune response? I think so, right? So if caloric restriction changes the microbiome, which changes the innate immune response, there's some kind of dual way interaction here, and that's going to take some stuff to suss out. Immunosenescence will affect the innate immune response. So we're going to turn that red, and we'll find the justification for that when we do the immune system. So immunosenescence changes this, and then maybe that goes and does a feedback. It would also change how caloric restriction works, because if caloric restriction is trying to improve the immune response by changing microbiome, and the immune response just degrades naturally, at some point, changing our microbiome isn't going to change the innate immune response. So maybe there's another secondary, we need to have more explanatory power. Okay. Ugh. Ugh. All right. So how did they measure? God. And then there's all the diagnostic tools, right? How do they, what tools do they use to even measure these things? Man, this is gonna be a lot, but it's very necessary. So we'll have to just go through it and just do it. Okay, so gut microbiota resides in the intestinal and coastal surfaces, digesting food. Microbe signals. Man, they're really atherosclerosis. P. So see, we need to talk to these people, and then it would be like 20 times faster if we just talk to these people. So Paul Wade, Stem Cell Biology Laboratory. Paul Wade. Hmm. He's not doing aging research. I mean, it's not about, I mean, it doesn't mean that we shouldn't talk to them, but if they're not doing aging research, opening it up to just talking to every single person in the field of microbiome is like, it's ridiculous. We could have basically a conference to, that was the idea. Like, we just start holding conferences for every single one of these sections, and we get them all in a room, and we get them to build this out and to really understand, okay, so what happens. Bone and brain, we're going to remove these for the time being. They're not specific enough. They're not specific pathologies. Alzheimer's is a pathology. Gingivitis is a pathology. Tonsil stones is a pathology. Parkinson's is a pathology. Centenary microbiome can go up here. Diabetes is a pathology. Obesity is a pathology. Psoriasis is a pathology. Eczema, allergies. These are all pathologies. And we're going to have antibiotics coming in. And then here we have measurement of centenary microbiome. Let's switch gears here and take a break mentally, but still work and do diagnostics. So measuring the aging microbiome. Healthy aging. Later, healthy aging. Microbiome clocks. Taxonomic functional signatures. I opened that in a new window, not a new tab. Jesus Christ. Okay. Multi-view learning. So we'll say we'll say that they built a clock, a microbiome clock. And what tools did they use to build this clock? Materials and methods. Um, okay, this is metadata. I don't, I'm not so interested in metadata methods. Um, rather about direct methods. So we'll put microbiome clock here as a diagnostic. And this is actually a diagnostic that comes out of the microbiome. It doesn't measure the microbiome. It is more of a it's an emerging diagnostic from the microbiome rather than something that is aimed at the microbiome. So we'll pull this out and we'll put it there, and then we'll turn this. Turn this purple, and we'll put this paper to this piece, and we'll turn this purple. And this is going to be used to, there's going to be a thing to shunt out of this. Like the microbiome clock is going to be used for something else later. So there's going to be a line emerging from here, but we want to look at how MALDI-TOF and stuff are used to measure the microbiome. So who is measuring the human gut microbiota? They did samples. Oh, this looks promising. These are samples were collected at home within 24 hours and transferred to 88C. Okay, this is looking cool. Frailty. That's on the people, not the microbiome. So 16S RNA sequencing. 16S, do we have that? No, we don't have that. So 16S RNA sequencing. 16S RNA-seq is used to measure the microbiome. There we go. Oh my God. Okay. Okay, 16S RNA-seq. And that's from this paper. And let's see if there's a PDF of it on here. There is. It's just got the most ridiculous link I've ever seen. So we'll use the other one. I mean, it's not the most ridiculous. Google image searches are the most ridiculous, but biological aging and the human gut microbiota. That's David Welsh from, okay, Vincent Maffei. Christopher Taylor, he's the correspondent. Maybe he has done other stuff as well. Let's just do a tangent here and take a look and see if we can find a lab of Christopher Taylor. Microbiome. Is he doing clinical interests? Productive, newborn, bacterial, vaginosis. Okay, so research interests among high throughput sequencing, human, so it's STD, microbiome stuff. Okay, not aging, so we don't need to. But I mean, he was part of this, but I guess somebody else, David Welsh, microbiome. What's his research? Host interactions, biological age, and geriatric syndrome pathogenesis. I think he would count. I think he would count. Let's throw his lab in with, what is this? Genomics resource group. Let's put his lab in with in the lab section. So Dave Welsh, LSU. So Welsh lab, and we're gonna do Phil Gray. Okay. Cool. That's that. So 16S, back to the topic at hand. We'll close some of these tabs that were from a very long time ago. Okay, microbiome. Biological aging in the human microbiota. So what they use? 16S RNA sequencing. Sample library normalization. So it was just 16S. Simple stuff. Okay. So see, so now we can say that this paper, and we'll use the other link to it, is a one instance. What that? Okay, yeah, so that's a paper. Did I put magnitude biosciences in the wrong? I did do that. Okay. Gotta check that these are correct. All right, and we'll save it as a graph file so that we don't lose our place, because this thing doesn't actually have a control Z built into it yet. So that's, disturbing. Okay. Let's see here, microbiome clock, antibiotics, MALDI-TOF. Let's find MALDI-TOF microbiome aging. The human gut microbiome in people with diarrhea, no. Mm. Uh, you know what? Let's use this one for the MALDI-TOF's existence. Mm. Microbial. Uh, and we'll just put it here. And we know that it's going to be connected to the microbiome. We just need to find what's it called? Something that shows. Oh, okay. Let's switch gears here again and just let's go back and take a look at the this thing in terms of microbiome and aging. So we can close that and we can close that. Okay. All right, this is a little simpler. And we can just go back and look at some core concepts here. Aging alters the microbiota. And why did they say that aging alters the microbiota? We got a little into the weeds there, but you know, that's the whole point of this, is to get into the weeds. Body stores modifications in the microbiota and flies linked to the agent has related onset of intestinal barrier dysfunction. Intestinal barrier dysfunction. We need a. Is that it? Is that going to tell us about intestinal barrier dysfunction? Now, the thing is, that happens in Drosophila, but it probably doesn't happen in humans. Let's I'll say that where we should probably 9 and 10. Let's just keep being surface level a little bit here. Aging of the human meta-organism. Oh, here's something by Franceschi. But let's see here. Aging alters the microbiota. Mechanisms that regulate host aging. What's today? Today's Tuesday. Okay. Regulate host aging. Regulate host aging. Okay. So this is what we want to see. This section here. So there's inflammation, immune system, and intestinal permeability. Leaky gut is the mechanisms, and we want to know how aging affects all these things, and then how these things are affected by aging. Okay. Or how these things affect aging, and then how aging affects these things. All right. So intestinal permeability, let's see. Aging and age-associated inflammation. The regenerative capacity of tissue diminishes. Aging has a deleterious effect on pluripotent stem here and the renewal differentiation abilities. This phenomenon has recently gut microbiome influences stem cell proliferation in the gastrointestinal tract. Regarding intestinal epithelial stem cells, studies in germ-free and antibiotic-treated mice revealed that gut bacteria influence the morphology. The morphology and function of the intestine. In the rodents is decrease the proliferation. However, the mechanism of how microbiota influences remains yet to be determined. Okay, so there's no mechanism, but here's some hypotheses. Stimulation of ISCs by uptake of the iron membrane in direct physical contact with the intestinal epithelium. So let's see. The microbiome, what's the domain of intestinal epithelial stem cells? Let's put that into, let's put that into, I want to put it into senescence, cellular senescence. We'll put cellular senescence into intestinal cell stem cell senescence, intestinal stem cell senescence, at which point we'll just drag this thing over up to the microbiome area. And we'll say intestinal stem cell senescence. So that's going to be a red because you don't want this to happen. And then this is going to be hitting the microbiome. But more specifically, it's going to be hitting the gut microbiome. Okay. And that's going to be red. Okay. And we can drag it all the way back. And the relationship here, let's just drop the microbiome and aging. Build that gut bacteria. So if we can get the gut microbiome influence on the mammalian intestinal stem cell niche, so we'll go to the source and we will find this, and we'll go here, and we'll take this and we'll put it here. So intestinal stem cell senescence then affects the microbiome, the gut microbiome. And so the pathologies here, the pathologies here are going to be tied partially to that. Okay, all right. But now see, now we have a train of logic where we have oncogene activation goes to replicative stress, which goes to cellular senescence, and it could be cellular senescence of the intestinal stem cells, at which point that comes all the way back up to the microbiome, and then it hits the microbiome, and then it'll spread out to the pathologies. So pathologies associated therein. You see, though, like how we're building these trains of causality, and you can like see how these things interact with each other. Well, at least from my point of view, they do. Okay. My God. Okay, we'll put that aside from now and go back to the original concepts. This phenomenon, this strong events. Okay. Okay. They go over some theories, but you know, molecular activity studies are required to confirm this mechanism. Studies of Drosophila also reveal the link between IESCs and the microbiota. So these metabolic impairments, intestinal barrier dysfunction to mortality during aging. So immune dysfunction, which is to say that's the immune system immunosenescence. There's probably, I mean, they don't describe it here, but I'm going to assume that that's what they mean. hyperproliferation, which is caused by, but it's getting created, they're creating more. That wouldn't be a senescence thing. That would be a genetic regulation thing. I'd have to look into it more. So this will be for the next, this will be for later. Microbial dysbiosis. Such physical alteration observed in annex flies. Alters digestive, short chain fatty acids. From inflammation. Innate immune system. In the gut microbiota structure. Okay, simultaneously aging research changes in the gut microbiota structure. Jesus Christ. There is a question whether there's a causal relationship between inflammation. There's studies of microbiome centenarians. Regulate cellular inflammation. So what they're saying here is that we have a microbiome population, immune response. Where's inflammation? Proteostasis, electrodynamics, metabolites. I think I might not have even put inflammation as a section yet. External organs, DNA. Nope. So let's do that. Let's create a new node and call it inflammation. And I think, and we'll create a child node within it called inflammaging. And I think that inflammation itself should just be yellow. The whole thing should just be yellow because it's a pathology. Well, we'll wait on that, I guess. I mean, is it? It's useful in normal operation. Whatever. I'll let somebody else argue about that. So microbiome is going to affect inflammaging. And it's going to affect it in a negative way. It's going to cause inflammaging. And inflammaging is going to negatively impact the innate immune response based upon this paper. Microbiome and Aging. So this paper is going to suggest that microbiome affects inflammaging, inflammaging affects the innate immune response, and that the innate immune response butyrate butyrate producing bacterial strains, Eubacterium limosum, have been identified in centenarians. So Clostridium, anti-inflammatory, interleukin, but metabolites, experimental. Can differentiate between commensal and pathogenic bacteria. Inhibition of JAK-STAT signaling prevents intestinal functional decline in life. Microbiota and the regulation of immunosenescence. Polyphenolic delayed immunosenescence through the modulation of microbiota. We're creating circular systems of logic here. But this is the first step, right? We're going to go through, we're going to build the spider web first. And then we got to go through and ask questions about the spider web and be like, oh, well, is it really like this? What's going on here? So we have a concept between microbiome and immunosenescence. Modulation of gut microbiota and delayed immunosenescence as results. Syringosinol consumption. This is mouse, but it's a mammal, so we're gonna throw it in there. Microbiome, inflammation, SIR treatment restores age-related dysregulation of lymphocyte subsets. Composition in middle-aged mites. Syringoresinol, polyphenolic lignan modulates immune aging in the gut microbiota associates. That's why our treatment delayed immunosenescence by enhancing the numbers of total CD3 cells. Well, first of all, let's see if this is even like statistically significant. Young, vehicle, SYR, CR is calorie restricted. Vehicle means they just put in the vehicle material, and SYR 10 and 50, I assume are different doses. 10 milligrams versus 50, or caloric restriction. Wow. So here's the vehicle. So this is like the cluster of them, and then this is the cluster. This data looks really good though, suspiciously good. Number of CD4 cells. It's like a little bit, but it's not a huge amount, and caloric restriction does more. But based upon prior knowledge, there's a belief that caloric restriction is partially acting through the microbiome. So it might be that they just didn't do high enough dose. But these other pieces, they look pretty good. Jesus Christ. At least these are what's it called? Okay. Flow cytometric, immunofluorescence, CD4, CD25. What do they use for the microbiome? Gut microbiota. (16S) again. Okay. Well, wait, hold on. Did we already put this in here? We did. And we said microbiome can induce immunosenescence. But immunosenescence just happens too, doesn't it? It happens just like even if your microbiome was perfect, it still immunosenescence would happen. This just could accelerate the process. Okay. And we'll get there. We'll put some stuff there. Yeah, we definitely need workshops. But it's all very interesting, so I will continue. Okay, multi-top. We will do purple here. Cell culturing is also used to measure the microbiome. Obviously, it'd be nice to have references, but it's like a really old method. I've actually done it myself. Gene sequencing, RNA-seq is going to be within gene sequencing. Then we have SDS page. I haven't seen an example of that yet, but once we do, then we'll put that in. Okay. Just to wrap this up, let's look for microbiome and Alzheimer's disease. Brain gut microbiota axis in Alzheimer's disease. Pathogenesis, Alzheimer's. So this is from the gut microbiome to Alzheimer's. We're going to put this link here. And I'm sure that there's more, but and I'm not sure how accurate this is. Oh my God, it's involving the enteric nervous system. Amyloids, lipopolysaccharides. Back to lipopolysaccharides and their involvement with the inflammation. Maybe it should just be a thing. Inflammation. Lipopolysaccharides. But someone with more experience than me is going to have to, I'll save as a graph file. Someone with more experience than me is going to have to suss all this out. Okay. LPS, that's great. Microbiome and aging. We're going to go back to this paper. Jesus Christ. I mean, we barely even got through it, right? So immune system inflammation. We have ties to both the immune system and to inflammation. Intestinal permeability is a function of those things. So it's a product of both of those. So we've basically covered this section. I mean, we haven't like specific covered everything in this section, but we have things referencing each of these sections. Host metabolism. So this is going to be dietary control. Transplantation of serum microbiota to native mice improves glycemic control and insulin sensitivity. Promotes beige fat development, reduces weight gain. So microbiome transplant, which is here, which actually, it would affect the microbiome and therefore affect glucose metabolism. Boom, boom. Let's do, that's going to be gut microbiome. So we're going to pull from gut to glucose. Oh no, we've already got one. Let's make this a non-directed link, and we'll put reduces weight gain 42. Let's put that functional gut microbiota remodeling contributing. Caloric restriction induced metabolic improvements. Did I already look at this? I think I did. Oh, this is the one without, okay. Okay. Oh my God, reactive oxygen species in here too. Post metabolism. There's some kind of mechanism for DNA methylation. Amyloid, inflammation, epigenetic alterations. Foxo. So autophagy. See, and this is why, like every field claims to be connected to every other field. Like they're the center of all existence, and it cannot be. You know what I mean? Like every field can't be the center of all existence. So there's got to be. It's like, let's map out these specific things that happen, and then we'll find. But here's the thing. So far, what about aging impacts the microbiome? We have the inflammation and the immune system, so immunosenescence. It's coming. There's nothing that's native. Does that make sense? There's nothing native about the microbiome that has a native problem associated with aging. It's all be coming from the outside in. It's coming from inflammation coming into the microbiome system and disrupting it. It's it's dietary control coming into the microbiome to change it. And whilst it does have these effects on all these other things, the question is, what is native? Why would the microbiome just change over time, well, as you age without any other input? It wouldn't, right? It wouldn't. There's nothing to say that. So their major cause is inflammation, which is an external factor that's coming from somewhere else. Dysbiosis, which could be cured by just, like, that's not an age-related thing. That would be a diet-related thing, right? Or it would be related to the immune system, immunosenescence, causing dysbiosis. So let's say that dysbiosis is the pathology that the immune system is going to affect. So whatever it comes in, we're gonna create a pathology here called dysbiosis, and we're going to have that negatively affect the entire. And we're going to put as justification, we'll just start with this as justification. I'm sure that there's better papers, but we'll start here. And what causes dysbiosis? Um, it's intestinal stem cell senescence does not cause dysbiosis. It'd be immunosenescence causing dysbiosis. This is supposedly failing microbiome causes immunosenescence, but immunosenescence causes dysbiosis, and immunosenescence is caused by other things as well. So we might have a small cycle, like a feedback cycle here. So might be an accelerant of immunosenescence, but it's definitely not. So let's say, okay, how would that work? The immune system is senescing from another reason entirely because it does this on its own from age. Um, then the immune system causes dysbiosis. Dysbiosis causes an increase in production of the epithelial cell wall cells, which creates like what stem cell exhaustion or something like that, which then induces more immunosenescence. Does that make sense? Does that ring a bell? How would you test for that feedback loop? I'm sure people have looked into this. I'm sure if we just hold a conference, we can. But this is a good question to ask. Okay, all right. Immune system. Let's delete this one because now we have a more specific set of connections between the immune system and the microbiome, and we don't need that generalized one. Um, okay. All right. Uh, is there any other core mechanism? We see mTOR mentioned here. That's still dietary control. Host protein homeostasis. Um, how does it affect proteostasis? Let's drag that thing over. DNA damage. Where's proteostasis? Here we go. Um, proteostasis is a subset of autophagy. Drag autophagy over here. And so it changes autophagy levels. Pollen produced by microbiota and a macroautophagy extends lifespan. Spermidine is produced by microbiota. Interesting. and a macroautophagy inducer, 40. That's interesting. Well, let's do this then. Let's go to macroautophagy. Let's do spermidine here. Let's fill that with this. Let's do this for that, and then um, microbiome induces spermidine. So we'll draw. Come on, there we go. Microbiome induces spermidine. Oops. So this is going to be for this connection here. But the idea that the microbiome produces spermidine, how does the microbiome produce spermidine? That's what microbe makes spermidine? The human gut bacteria, bacteroids, and feels some produce putrin spermidine and system pectin. No, bio. We'll use this. We'll use this. Come on, Oxford. Yes. Okay. Oh, PDF. Is it PDF online? It is online. It's got a ridiculous thing though. So okay. So microbiome naturally produces spermidine, which then induces macroautophagy. Jesus Christ. This is why we need to go through this stuff. Oh God, my back. I'm leaning over too much. And it's just about the top of the second hour. So let's go through this really quickly. What else is here? Protein homeostasis. They're talking about heat shock proteins. Holy shit. DNA maintenance, telomeres, epigenetic. God damn. We really do need like a conference on this whole thing. Model organisms. Of course, they talk about C. elegans, fruit flies, and all this other. They're just killifish. Find another animal. Holy shit. You ever heard that joke of like people who talk about Harry Potter too much and people just shout at them like, read another book. Like it's just like, find another animal. Stop looking at these flies and worms. It's like, dude, there's other animals out there. Come on. Oh, okay. But structural molecular biology. We should get in touch with this guy. We should find out who he is. Because he's probably doing microbial aging. And yes, he is. So this is a good lab to add. This is a good lab to add. The Cabrera lab. Put a link there. This is fantastic. Okay. Jesus Christ. Let's throw this back over here. Let's throw this here for now. Okay. Okay. Ah, Jesus. And we'll just turn this turquoise. Okay, that's it for me. Holy crap. Stop share.